Hello again. So today I thought we would take a look at something called tissue fluid. Now, from GCSE and before, we know that substances move from blood vessels, the capillaries, into body cells. So that could be anything: glucose, oxygen, water, mineral ions, amino acids, fatty acids, any of those things. But what we don't consider often is this space here. So the space between the capillaries surrounding all mammalian cells. And that is filled by something called tissue fluid. And that's really, really important because what actually happens is these substances move from the blood vessel into the tissue fluid and then from the tissue fluid into the body cell. And we're going to look at how this tissue fluid is formed. So if we think about how tissue fluid is formed, so first of all, we have blood entering from an arterial end into a capillary bed. And the blood at this arterial end is under high pressure. It's called high hydrostatic pressure. And that means hydrostatic pressure. And that's the pressure caused when fluid is forced out of the capillaries. So due to the heart beating, this pressure causes water and small molecules to be forced out of the capillaries through tiny holes between the different endothelial cells into this yellow surrounding tissue fluid, which is bathing the cells. Proteins and red blood cells are too big, so stay inside the capillary. So only water and small molecules can move out into the yellow tissue fluid on my diagram, whereas proteins and red blood cells stay within the capillaries. And this process is very similar to the process going on in the kidney in ultrafiltration and is also called ultrafiltration. So that's how tissue fluid is formed. We also need to consider the other end of the capillary bed. And as blood travels through this capillary bed, the hydrostatic pressure reduces because we're getting further away from the heart and some water has been lost from the capillaries into the tissue fluid, reducing that hydrostatic pressure. The proteins within the blood vessels, the proteins in here, were not able to move across to the tissue fluid due to being too big, exert a special kind of osmotic pressure called oncotic pressure. Okay, that oncotic pressure. And what that means is the water potential inside the capillary is more negative than that in the tissue fluid. So we've got a lower water potential here than we have in here. And what that means is we are going to get the movement of water along that water potential gradient back into the capillaries from the tissue fluid. If you have certain problems like high blood pressure, then that can mean more water being forced out. The hydrostatic pressure is higher. More water is forced out into the tissue fluid, less water returns back into the capillaries at this end, and therefore you'll get a buildup of tissue fluid, otherwise known as something called edema. So if we now look specifically at the pressure changes, we can see that as the proteins that exert this oncotic pressure, so I'm going to oncotic pressure, remain in the blood. So this is the pressure in the capillary as we go along from the arterial end to the venous end. The oncotic pressure exerted by the proteins remains the same within that blood vessel. And that's because the proteins, notably usually it's albumin's the main one we're talking about, do not move out of that blood vessel. So that oncotic pressure stays exactly the same. We've talked before about the hydrostatic pressure. So that's the pressure that is exerted. So hydrostatic pressure is the pressure that's exerted on the fluid within the capillary. So it's due to the heart beating, so forcing water and small molecules out of the capillary. And that starts off really high in the arterial end, really high, but it decreases, partly because we're getting further away from the heart, but also because water and small mineral ions are moving out of the blood into the tissue fluid. And that means we get a change in pressure. So where the hydrostatic pressure is greater than the oncotic pressure in the blood vessel, we're going to get water and small mineral ions and other molecules leaving the capillary. They're moving out into the tissue fluid. When the hydrostatic pressure decreases and falls below the pressure exerted by the proteins within the blood vessels, we're going to get water moving back into the blood vessels from the tissue fluid. So sometimes we might hear the term filtration pressure, and that's the resultant pressure. So for example, at the start, so if we think about filtration pressures, so at the start, the hydrostatic pressure could be, for example, 30, okay? The oncotic pressure is going to be drawing the opposite way to the hydrostatic pressure. So we're going to give it a negative value, and let's say that that's 25. The overall filtration pressure, therefore, is plus 5 at the start of the capillary. So plus 5 overall filtration pressure, forcing blood and small molecules out from the capillary into the tissue fluid. But as I've mentioned, as we move along the capillary to the venous end, the hydrostatic pressure will decrease. So let's say it decreases to 20. The oncotic pressure, this osmotic pressure caused by the proteins, will remain the same at minus 25 because the proteins aren't going anywhere. They're too big to move through the endothelial walls. And so overall, this filtration pressure is now minus 5. So it's going in the opposite direction. So in this case, we've got the oncotic pressure due to that low water pressure, low water potential, drawing water back into the capillary from the tissue fluid. Other products in that tissue fluid would also be waste products, so things like urea and carbon dioxide, waste products from the cell. So we've talked about the capillaries and the movement of water and small molecules both into the tissue fluid and out of it from the capillaries, but that's not the whole story. So not all the tissue fluid returns to the capillaries. Some enter something called the lymphatic system via lymph capillaries or lymphatic capillaries. So it bypasses on the way back the venous drainage and goes into the lymphatic system. So fluid will move along these lymph vessels as they're compressed by body movement. So there's not the same heart beating, forcing the fluid around, but contractions of body muscles and body movements will cause this fluid to move along the lymphatic vessels. And they do, similar to veins, have valves that prevent backflow. And the lymph will eventually drain back into the blood near the heart. And it's really important in transporting some things like fatty acids from the digestion of lipids, as well as some white blood cells, so lymphocytes. So let's take a look at the sort of question you might get asked. So in this one, we're told blood is pumped around the body in blood vessels to supply cells with nutrients and oxygen. You're first asked a nice introductory question where you're asked to fill in the table and show where endothelial cells and valves are found. So endothelial cells, hopefully we know, are both capillaries and veins. Valves, though, are veins only. And then we're asked, nutrients and oxygen pass into cells from tissue fluid. Explain how tissue fluid is formed. Well, from what we've gone through, hopefully you now know that fluid from the blood, fluid is forced through the capillary or you might even say endothelial wall or endothelial cells, due to the high hydrostatic pressure. And that's obviously at the arterial end. So nice, easy questions to answer once you understand the formation of tissue fluid and how it also returns back to the blood